Procedures on the chemistry practical, identifying the different types of carbohydrates. Materials. Different carbohydrate samples, for example, glucose, fructose, sucrose, starch, cellulose. Benedict's solution or Fehling solution. Beaker. Test tube. Heat source. Pipette. Dropper. Procedure. Place a small amount of the carbohydrate sample in a test tube. Add a few drops of Benedict's solution or Fehling solution to the test tube. Heat the test tube over a heat source until the solution boils. Observe the color of the solution. Results. If the solution turns red or orange, then the carbohydrate is a reducing sugar. Reducing sugars are carbohydrates that have a free aldehyde or ketone group. If the solution remains blue, then the carbohydrate is not a reducing sugar. Non-reducing sugars are carbohydrates that do not have a free aldehyde or ketone group. Example: carbohydrate sample. Benedict's solution or Fehling solution. Result: glucose, red or orange, reducing sugar. Fructose, red or orange, reducing sugar. Sucrose, blue, non-reducing sugar. Starch, blue, non-reducing sugar. Cellulose, blue, non-reducing sugar. Drive spreadsheet export to sheets. Safety precautions. Be careful when handling hot test tubes. Wear safety goggles when working with chemicals. Tips. If you are using Benedict's solution, be careful not to overheat the solution, as this can cause it to decompose. If you are using Fehling's solution, be careful not to add too much of the solution to the test tube, as this can cause the solution to precipitate. If you are unsure whether a carbohydrate is a reducing sugar or a non-reducing sugar, you can perform a secondary test, such as the Barfod test or the Biles test. Applications. Identifying the different types of carbohydrates is important in the food industry, as different carbohydrates have different properties. For example, sucrose is a sweetener, while starch is a thickening agent. Identifying the different types of carbohydrates is also important in the medical field, as certain carbohydrates can be used to diagnose diseases. For example, the presence of glucose in the urine can be a sign of diabetes.